So I'm going to tell you a little bit about a conference that's just happened called ECCMID. I'm not going to be too technical about anything, so it shouldn't be too dry, hopefully. So what's ECCMID, ESCMID? It's a really huge medical conference, and it's about all the different infectious diseases. So bacteria, fungi, viruses, weird parasites, some of which I've never even heard of. And it's brings together doctors and researchers from all across Europe and some from beyond. And it's a really good platform for some of the really important things that need to get discussed, because you have such a lot of people in the audience that you get really insightful questions. So it's quite an exciting one. It's overwhelming. There's more talks than you could ever watch. I think a lot of the people that went, they're still watching the recordings just to catch up, because there'll be like 10 different lectures happening all at the same time, and it was for 4 days, so huge amount of content, but some really exciting stuff got presented. So we didn't have any we weren't presenting any Aspergillus stuff this time, but the mycology lab that NAC has, MRCM, they had some very important new work on a PCR test that they do for a different fungus. So it's called pneumocystis. It's not a close relative of Aspergillus, but it has a lot of the same, there's a lot of the same kind of difficulties and challenges in the diagnosis. So we're in quite a good position to be able to work on this. So things like one of the questions is, as with Aspergillus, when you measure how much of the fungus is in the lungs, how do you know if it's just a little bit, it's colonized, a little bit is living there kind of harmlessly, and how do you know when it's an infection that needs to be treated now? And that has been quite tricky for pneumocystis, and it's been quite controversial. So the NHS uses the PCR test now, but in America, there's a lot of people who sort of say, oh no, it's not trustworthy enough. And what happens is people end up being put on very strong medications, and it turns out afterwards that they didn't actually have the infection, and they didn't need to take these really toxic drugs. So some people from the lab, so in the picture you see Rena, she's like the head of the lab. They did a massive study. They pulled together like all the data that exists, all the good quality data from like 10 years, and did a really strong statistical analysis, put it all together, and really proved once and for all this is a good test. And also you can get like extra mileage out of it by doing, you probably heard the beta-D-glucan test. So that's one of the fungal antigen tests that you can find in the blood. If you combine the two of them together, then you get even better results. And that's something that isn't done in the NHS often enough. So there's going to be a big paper coming out soon, and we're going to do quite a lot of promotion and probably start some arguments on Twitter with some American doctors, but we're going to win the arguments. So that's really exciting. And she did a really good talk, it's difficult when it's about statistics, like how many people really want to sit and hear about difficult maths for an hour. It's difficult to make that fun, but somehow she did. And her PhD student presented some of his PhD data. He's on the right. That wasn't about mole stuff. Oops, sorry. There was an art competition, which is also pretty exciting. It seems to be getting more common in science stuff these days. So my absolute favourite one on the top right, that's an agar plate. So somebody's made Hokusai's Great Wave. They've got an agar plate, and they've got different types of bacteria that grow in different colours, and they've painted it onto the agar and let it all grow out to make a little tiny living painting. And I just love this stuff. It's so good. It's so good. So yeah, I'm going to just tell you a little bit about three presentations that I thought were very cool. I wish I could tell you, as ever, that there's some great new antifungal that we're presenting for the first time, and we'll be able to give you all it next week. Obviously, that it just isn't possible. So I know it can be a little bit frustrating here about the sort of incremental little baby steps, but it's what we've got. So one pretty cool thing. So olorifim, you'll have heard us talking about. It's in phase 3 trials, and some people with very severe infections are starting to be able to get it on. They call it compassionate use. It's where somebody's just absolutely run out of options. So at this stage, that is kind of on the cusp of being used for invasive infections. And I think the data that F2G, the company that makes it, presented this time, it shows they really are serious about using this for Aspergillus. So what they did was, so as well as the clinical trials, they also do a lot of like laboratory behind the scenes work as well, in vitro stuff. So what they've done is they got almost 7,000 Aspergillus isolates from patients all over the world. I mean, 7,000, that is massive. And they tested olorifim against them to see, like, they already know it works against Aspergillus in principle, but what they wanted to know is, are there people out there, is there much resistance out there? Is this going to be a problem coming up? And so, I mean, this, like 7,000 isolates, this is exhaustive. That is absolutely astonishing. And almost everything was susceptible. There was almost no resistance whatsoever, which is fantastic news. And this covers lots of different countries as well.
So were these isolates resistant to other antifungals, Beth?
Some of them were. I couldn't tell you what proportion off the top of my head. Yes, yeah, quite a few, I think, were azole resistant.
So what we're getting here is that there are probably 1,000 or so patients who have got resistant infections, so we can't treat them at the moment. Olorifim will potentially be able to treat those people.
Yeah.
That's fantastic.
And also, it means that there isn't. It's not that it's not going to come to market and then we're going to find out, oh, actually there was a resistance gene that was in circulation that we didn't know about, and it's just suddenly spread. It means that, yeah, the chance of it, things becoming resistant quickly is low. It's a really good sign, I think. And just the amount of work that went into that, because I think the number of strains of any fungus they already had on the books was like 9,000. So this is almost coming up to the same again, just Aspergillus, and different species of Aspergillus as well, which was really good.
I presume someone had to plate all of those out and watch them to see if they grew, rather than them being a machine doing it all for them.
I think it probably was manual. There were other talks that I'm not going to get into now at ECCMID where they were using AI and photographs of plates growing, and actually in the early stages where they weren't quite sure what it was going to turn out to be, actually the AI in the photographs, it did better than a human, which is pretty cool.
Yeah, maybe we should talk about that another time.
Yeah, I think there was 3 different talks, and I want to watch them all before I say anything about it, because it's not something I know that much about. But
just interesting fact, F2G was founded by David Denning, amongst other people.
Yeah, so
it's a Manchester company, so we have a big stake in it.
Yeah, and it's tantalizing that it's getting so close to market. It's like to being more widely available. It's very cool. Yes. Okay. Yeah. So that was a little step forward on that one. Another one that it's not anywhere near that stage of development, but I just thought it was a really interesting approach, is at the top there's a group in Aberdeen, and they're developing an antibody therapy that it's a bit different to other antibody or immune therapies that we think of with Aspergillus. So we normally think of, so like biologics for ABPA is one kind, or like interferon gamma sort of boosting the immune system. But what they're looking at is can we get an antibody that sticks onto fungi, just sticks onto the surface, and then our own macrophages can see them and go and gobble them all up. Basically, like putting loads of little tiny flags on the fungi so the body knows where to go to get rid of it, which is absolutely amazing idea. So the work, it's an early stage. So they're using Candida yeast at the moment because it's just easier to grow in the lab. You just grow it in a flask, it's much easier. But if it works well, that might be something that they then expand for other more difficult fungal species like Aspergillus. So they've done a really nice body of work. Like they selected all they grew up the yeast and saw what changed in the yeast when they started being infectious, and then they picked all of those out as the targets, and then they designed different antibodies, and they tried the antibodies out in mice. So you give them mice an invasive Candida infection into its bloodstream, and the antibody gave 83% better survival, which is astonishing numbers, and especially for something that at such an early stage and such a simple sort of neat idea. So that will be something that will definitely want to be following up on. That's Carol Munro, you might know the name.
Yes, I do.
Yeah, that's her group. So that's super cool.
You would expect our bodies to make antibodies like that to attack the Candida. I presume this is more targeted or something like that. That's why it's working better.
I think in this case it's more like in an invasive infection, then the body doesn't have time to produce.
Yeah,
it doesn't have time to generate its own antibodies. And also I think because Candida is something that's naturally present on the body a lot of the time. You can get used to it as part of your flora. And also, like, sometimes some things are quite difficult. Some things aren't very antigenic.
Yeah, fungi in general are quite good at avoiding detection, aren't they?
Yeah, the chitin is quite immunosuppressive.
So this may be the other angle of that is this may be the basis of a future vaccine, perhaps, or something like that.
I don't know how it would tie in with that. I can look
Just identifying regions that a vaccine could also target, which would be able to pick out virulent Candida or virulent other fungi.
Yeah. Just the epitope. Yeah. Having a list of
that's really good. Yeah, that's cool. A lot more than it sounds. That's really very useful, isn't it?
Yeah, and it's a completely different type of treatment, and it shouldn't be anywhere near as toxic as like a chemical.
And we know these macrophages attack and kill Aspergillus as well. So the same approach could work also for Aspergillus and other filamentous fungi.
Yeah, I might send her an email and ask what the plan is, if she's got any plans to look at Aspergillus. Because if she has, that could maybe next year, when it's a bit more developed, we could have her as a speaker.
She was the president of the BSMM, so I'm not sure whether she still is.
I don't know. Yeah.
We did, yeah. We did some work for her about 5 years ago.
Oh yeah. Yeah, so that's quite an exciting one.
Very nice.
And the third one that I'm not going to get into any level of detail. It's just an interesting idea that we don't talk about that often. So there's an Italian group and they're looking at, they call it repurposing drugs. So it's where groups, they take a medication that it's already on the market. We know loads of information about the safety and the manufacturing. All of that development's already been done, which saves a huge cost. And then they try and see if it does. Yeah. So they're trying to see if it will if they do other things. But this is a bit of a funny one, because the drug they're looking at repurposing, it's called vortioxetine. It's an antidepressant, so that's a completely different type of disease. So you would never, it's not an obvious choice. So I'm not quite sure where they got that idea from, but it does seem to be antifungal, which is quite a funny thing. So again, it's at a very, very early stage, but it did, when they applied it to fungi, it did cause them, that they call oxidative stress and death in the fungi. So if it turns out that that's a reasonably effective antifungal, that's one that could be fast tracked into giving it to patients. That'll be a lot quicker than developing something from scratch. And also it's quite a, I'm not sure what the mechanism is, but the structure looks completely different to other things. So that would be a whole different, like it's not similar to stuff. It's not just more of the same of what we've already got. It's completely different, which is good for things like resistance or combining it with other things.
So an example of just letting the fungus decide what it doesn't like, rather than us trying to think of a way
Yeah.
to design a molecule. These big drug companies have chemical banks of probably millions of different chemicals, that a lot of which would be based around drugs that they've developed, and they just test everything. They do it with big machines, big robots. So unlike Rena's work, they could probably do 1 million in a week.
Oh yeah.
And this is the kind of result they get out of it, which could be really useful. It could be, we're always saying it's 5 years, 10 years away from clinic, but something like this would only be 3-4 years away, because all of the safety data has been done on it. And in fact, it's been in use in the human population for some time. So it would be really good. There's lots of surprising. I know, I remember itraconazole was being looked at to treat cancer at one point.
Oh yeah, I don't know what happened with that. Yeah.
Probably wasn't effective enough. Again, they'll test it on a real group of real animals or people, and just see whether it had actually any effect in the real world, rather than trying to do it in the lab, I would imagine.
Yeah, I think it's really good for making clinical trials easier, because like when you look at, say, fungal infections, it's so broad, like comparing invasive aspergillosis to even chronic forms. They're such different diseases that you might end up finding that there's a drug that works for one, but one fungus, but not for another. And if you can just make the clinical trials a lot easier, it makes them possible.
Yes,
so we can try out more things.
And it can be for completely unexpected reasons that something will work or something won't work. So best not to try and second guess everything.
Yeah.
Very good.
One to keep an eye on.
Right. I assume everyone can see that.
Yep.
And hear me. Just this is going to be really quick. Respiratory viruses have disappeared. Let's just say, with COVID, which is a thing we've been talking about for 3 years. Is it 3 years? It's 4 years now. Might be 4 years. We were no longer collecting very much up-to-date information, because the risk to human health, I suppose, has dropped somewhat, so the lack of urgency. But I did find this graph on the main dashboard for COVID cases from around this time last year. And this is cases. This before I've showed people being admitted to hospital or people dying from COVID. Well, those things, that data now stops before the beginning of April, which isn't much good to us. So this is cases detected, and it's not a perfect measure, because you'll only detect these as you find them. So as we're not actually looking for these for COVID anymore in the general population, these are probably a bit on the low side. But what I'm getting at is, just look at the shape of the surges that we had this time last year, over the summer into the autumn, and this one we had just coming up to New Year. It's much smaller, each time much smaller. But since end of February, absolutely flatlined. So I think that's a reasonably strong suggestion that we haven't had a surge in COVID, despite quite a few people I know of.
Yeah, that's weird, because I know more people with it now than I think I ever have.
But have they got it, or do they just think they have it?
No. The people I know they're all kind of dotted about healthcare, so they've all tested.
Right. Well,
yeah.
This, and I have to say, the data I usually show you shows pretty much the same pattern. It stops there, but it's flat also in their pattern. So maybe in that data, when they release more information, we might be seeing a surge. But as far as this goes, that's pretty flat. There's still plenty there. That's still looking at 200 cases a day, I think you're looking at there, that are detected, that are recorded. And it could easily be there's more around. But yeah, certainly looks like there's no. The COVID is continuing the trend of becoming less troublesome to us. Hopefully those people you're talking about, Lauren, didn't end up in hospital. I know the couple of people I know of that think they've had COVID certainly didn't end up in hospital, but they stayed in bed for a day or two. So it was bad enough. But it certainly looks like the trend for it disappearing is continuing. Which of course is fascinating for people who study viruses, because they've never had this opportunity before at this scale. We have figures for the 1920 flu epidemic. We have some figures for that, but nothing like this level of detail. But overall, it shows the same pattern where the virus starts off very damaging, and as we adapt to it, and it adapts to us, the overall effect becomes less and less troublesome in terms of hurting people's health. So pretty good news. It is continues to disappear
at the moment, isn't it?
It's what, sorry?
The one that spread my lad and all his family, they've all had whooping cough.
It could be. I don't think
which is quite prevalent in North Kent, apparently.
Right. I think we had, again, it's people who think they've had a whooping cough. We had lots of people coughing between summer and the end of the year last year here in work and various other places. So a lot of people then were saying that was whooping cough, and there was a surge in whooping cough. I don't know if that's gone down again. I can have a look at that before the end of this today if I get time. So that looks good. Similar sort of advice. If the vast majority of people who are poorly with this virus or are ending up in hospital are quite elderly. So if you are age 75 and over and you are offered the vaccine, then you should take them up on that offer because it's still out there. It's getting harder and harder to actually come into contact with, theoretically, but they are offering a spring booster vaccine. And there's a van at the end of my road which says COVID vaccine van, and it's not moved in 4 weeks. So I don't think it's being very active, but it is out there. You can go and get it. If you've been offered it before, you may well be offered it again, and it is still, if you are in a high risk group, something you should go for. I have still not seen any information that they're going to offer it to healthcare staff this time. I've certainly not seen it. So maybe that's being rolled back, and the actual risk to having lots of people off work is subsiding rapidly. So that's also good news. There's no evidence of any surge that we can find, or that I can find. Risk of hospitalization is going down, very low. If concerned, if you are elderly, for example, over 75, you can still protect yourself in all the usual ways. But do remember that socializing and walking and talking to people and making contact is an important part of your health, and get vaccinated when called. Right. Now, I'm going to try and do a talk on drug interactions and why antifungal drugs are particularly prone to this sort of thing. We thought about doing this a few times, but it is very complicated. And I certainly only understand about 1% of this subject. So I'm going to give it a shot and see how we get on. I hope to emphasize certain bits which explain why we emphasize so much that the drugs that you're taking, the antifungal drugs, are so prone to these interactions. This paper has just come out from David Denning. He's still out there working away, and Sarah is the pharmacist, specialist pharmacist that I worked with for quite a long time. And Russell Lewis is actually an American who works on CF children, I think. Anyway, there's quite a few big names in there. Andreas Groll is quite a big name in medical mycology as well. And what they're talking about are these antifungal drug interactions caused by the triazole antifungal drugs. So all those drugs that we have that end in azole fall into this category. We've had what they're talking about is mostly the antifungal drug interactions database that I developed along with a student way back in about 2012. That's carried on, and they've developed it, and it is available online at aspidillus.org.uk/antifungal drug interactions. You can also find it, we link out to it from aspergillosis.org. It's important. The original idea for this was to give patients a chance to check whether their drugs they're taking are prone to interacting with the antifungal drugs that we were giving them. And this is usually a good idea to support the doctor and the pharmacist in deciding what drugs to give you. To have something like this that patients can check themselves, because only the patient knows. Well, the pharmacist should know, the doctor should know, but the patient is more intimate with all the drugs that they're taking. And using this app, they can look up any antifungal drug and any of the other drugs they're on and see if there's an interaction. And some of these, the app shows the drugs being the interactions in a number of categories. You've got severe and moderate, which I'll show you in a sec. Severe essentially means life-threatening. So those are the things that doctors absolutely avoid. And comfortingly, those are the interactions they will most be looking out for. Let's see what the next slide says. Right, I'm just talking about, there's the interaction database web address, antifungalinteractions.org. There was an issue over the last few months where it would only work if you put www. At the front, but that's now been resolved. And it's very simple. You go there. There's a big box where you can search for any drug that you've got, and it has to be the drug name rather than the brand name. So it has to be itraconazole rather than Sporanax, for example. And a big long list will pop up as you start typing with suggestions for what you're looking for. You can click on that, and then it will pop up with a complete list of all the possible interactions of that drug with all the antifungal drugs that are available on the market, and it will tell you whether they are severe, medium, or otherwise in sort of red, orange, and green colors. So like this, red, that's more yellow, but that's supposed to be orange, green colors. This is important, just to give you some idea. I think we're covering most of these antifungals now. So you've got everything from Ambizone, caspofungin that we don't use an awful lot. You've also got posaconazole, itraconazole, voriconazole are all there. What sort of tendency, what are the severities of the interactions that they potentially will cause? Something like caspofungin, we don't worry too much about, because that only causes 6 severe interactions. Whereas if you go up to posaconazole or itraconazole or voriconazole, you can see there's much higher numbers of severe interactions with important drugs. So this is why we've made so much effort to produce this information. There's much, as you can see, there's also moderate, mild, and unlikely. Unlikely means we know a lot about the structure of the drug we're talking about, and we don't think it'll cause a problem because the interactions severity depends on what the drug's being used for, also the structure of the drug. And unlikely means, well, this is not a structure that we're worried about. And the vast majority of drugs fall into that category, as you can see. But when we see hundreds in the sort of high, moderate and mild, then we start to say, you really need this information in your hand to make your own decisions, as well as the doctors and the pharmacists. Pharmacists, no way can a pharmacist remember all of these interactions themselves. So on the same app, you click a button and it switches over to a version of the database that's for use by professional pharmacists and doctors. To start the story, this is what leads to this range of interactions. You've got your polypharmacy, so that you're taking more than one drug. So obviously you have to take more than one in order for there to be an interaction. Very much depends on the perpetrator drug, which is one of the two, and the victim drug, which is the other one of the two. They have particular characteristics, and they're important for the severity of the interaction. Inflammation plays a big part, and this is getting into the more of the subject of today. Patient genetics plays a big part. So in the same patient, we may get more potential for interaction compared with other patients, and it's the same as one patient may be more prone to side effects than another patient. So patient genetic variability plays a big part, and this is why you will see researchers now speculating that we need, and have been doing for 5 or 10 years now, we need personalized medicine, because these drugs interact with us in quite specific ways, and in some people there'll be a problem, in other people there won't be a problem. In some people it might work better, and other people it might not work very well at all. And if we examine what's causing that, and possibly by looking into their DNA, we can find out which one to give each patient. It's sort of personalized medicine. Naturally, when it was first mooted, sequencing DNA was very expensive, and so we couldn't proceed doing that in clinic. But nowadays, there are entire genomic units at several NHS hospitals. I don't know whether they've all got one, but I know MFT have a patient genomics centre now, and the ultimate aim of that is to be able to personalise the way we treat each patient. I don't think they're anywhere near that yet, but that's the aim. And other things you've got, as you all know, if you all have aspergillosis, that can profoundly affect the types of interactions and the severity of interactions, because you've got other stuff wrong with you, and I'll talk about that in a sec. So that's why, the reason why we're doing this sort of thing. Just to give you an illustration, this is the very severe end of these interactions. These are not things that you will ever come across, I don't think, unless of course you have a heart condition, which is quite possible. These are things that doctors will know to absolutely avoid. These things, if we give you all triazoles with this drug, all triazoles with this drug except for 1 or 2, what we get is an increased dose compared to what we were hoping for in those patients. And that counts for all of these. Increased risk of QT prolongation, well, that's a heartbeat issue. And the reason we have an increased risk is because taking fluconazole increases the blood levels of apomorphine. And the same with these other, I won't attempt to say that quetiapine. It interferes and it increases the dose in your blood, which is not good when when it's controlling heart rhythm. We've got to keep those drug levels under better control than that. However, if you go down towards the bottom, rifampicin, which is a drug I know quite well, and we use it a lot in science, or used to, will interact with any of the triazoles, and you will get an accelerated metabolism of azole, so of the high dose posaconazole. In this case, rifampicin causes posaconazole to the drug level to drop. So we might be giving you posaconazole and it might be having no impact because you're also taking rifampicin, and the actual levels of posaconazole in your blood might not be high enough to have that effect. And that applies to all azoles when given with rifampicin. And that's a problem because I think I'm right in saying is rifampicin is used for tuberculosis. And tuberculosis is something that makes you prone to having aspergillosis, especially CPA. So you might come across a situation where someone with TB, they attempt to treat them with an azole, and really that would be quite difficult because of rifampicin's impact on the triazole. Same with others.
Fentanyl's quite interesting, isn't it? That's used quite a lot in emergency medicine.
Yes.
Paramedics and A&Es dishing it out there. And do they ask if you're on some?
Well, exactly. They need to. This is the sort of thing they need to know, and you don't know that they need to know. I would imagine. So that's
just to butt in quickly.
Yeah, carry on. Yeah, we've got plenty of time.
Yeah, there's some people where it's even worse than that, because say if somebody's having a seizure or they're just in some other state where they're not able to respond at all, actually some seizure medications are very bad for interacting with azoles, and if you're not conscious you can't tell somebody not to give you it, and it's so urgent they can't just not do it. So that's when we were doing the wallet cards, it used to say on there, "Don't give me carbamazepine, please." Okay, that's all I have to say.
Do you think in those situations, if someone ends up in A&E, they're obviously not going to take any further dose, and because you're not allowed to take your medications into hospital subsequently, maybe the sort of protection from that is that's the protection from falling into that trap, because there'll be less azole around as time goes on and less impact, perhaps. I'm just trying to think safety wise.
Realistically, probably the person's going to come round fairly quickly. It's just more if they have a very acute reaction.
Right.
Like the chance of that happening, obviously, it's not something that's going to come up often. But
I would imagine sorry, you carry on.
When I went in a few weeks ago, I took all my meds with me.
That's
good. And they then dished them out. The nurse, when she did her drugs run, she gave me my own meds back.
Right.
But they were giving me itraconazole and omeprazole at the same time. Which of course is a no-no.
It's a no-no if you normally take them differently. If you've always taken them together, you might be put on a higher dose to overcome the acid effect, if you've always done it that way, but more usually you would take them separately.
Now I take them separately. Yeah, as soon as I got home, I
it's a lot cheaper as well.
Separately, yeah.
Yeah. So cause lots of hazards, these azoles. I'm very interested to know what we know about the new drug that Beth was just talking about, and now maybe that will actually be more. Another reason it might be more useful is it's not an azole, so it won't be causing this huge range of problems.
Yeah.
So what is going on? What is causing all these interactions, and what is an interaction? Now this is massively complicated, but as you can see, the PowerPoint these days actually offers you help. And you start typing in a word like pharmacokinetics, it actually offers you a nice image like this to go with your text completely automatically. So my slides are now more entertaining, as you can see.
Cool day, aren't
So the key word is pharmacokinetics, which you've probably heard us say a few times. Pharma, drug, kinetic, movement. So it's just saying moving drugs around the body, essentially. To work out what the what interactions are, we have to do a little bit about pharmacokinetics. So it's just, I'm going to call it the journey through our bodies of every drug that we take. And that journey starts with absorption. So there's the medicine, we take it into our mouth and it passes down into our, back. To our tummy, and it's absorbed into the gut and so on. But of course, there are other, I will talk about it slightly a bit more in a minute. So absorption, how does the drug get in? Distribution, how does it get around the body? Obviously, mostly in blood, but I'll talk about that in a minute. Then how is the drug doesn't stay in our bodies forever? Why does it not stay in our bodies forever? Why do we need to take more than one tablet every day? We have to take them every day. We have to take, if we want to stay on that drug, we have to keep taking it, which is good news for the pharmacology companies, but not so good news for us because we have to keep paying for the things. Why is that? Well, that's the drug is metabolized. I'll talk about that in a sec. And then ultimately it's excreted in the normal way, and that's through urine or through feces and so on. So that's the journey through the body, and that's what pharmacokinetics studies. It's literally the science of taking medication and how it impacts on our bodies. So starting at the start, absorption. So you've taken this drug, it's gone into your gut, into your tummy, down into your gut, and it has to be absorbed through the gut in some way. Other options are you can inhale. Obviously, we all know about inhalers. In that case, you're inhaling a gas which is absorbed through similar cells, but they're the cells lining your lungs instead of lining your gut. And there are a number of other ways. IV, of course, bypasses these mechanisms because it's an injection directly into your bloodstream. So there is no problem with absorption because it's not absorbed, it's injected directly. This peculiar figure is showing you the surface of a human cell. It's also a bacterial cell, but mostly a human cell, as if you were looking at it side on. So here's the outside surface of the cell membrane of your cells. So this out here could be the surface of your areas of your lungs, which would have a layer of mucus along the top. And in here is the inner surface of your cell, and that's where we want to get the drug. We want it to be absorbed into the cells underneath this surface layer. And this surface layer is quite a barrier. Obviously, on our skin, there's loads of layers of tough protective skin cells above this, and that stops anything getting in pretty much. But in our lungs and in our gut, that doesn't exist. We've got this much more delicate, and it has to be more delicate because we want to get substances through it. And if the substance we want to get through it is what we call, it's literally, is it soluble in fat? Because these little objects are fat molecules, essentially. And if you've got, if one of these was the drug we're trying to get absorbed, if that dissolves in fat, it will simply lie on the top, dissolve into the fat, and naturally pass through inside the cell. So a fat soluble molecule gets in, no problem at all. It's just like if you, well, what's a diffusion example? If you've got a lot of salt on one side, you've got a semi-permeable membrane like a cell membrane, and then not much salt on the other side. Naturally, these molecules will flow in one direction into the cell. That's the simplest and easiest way to do it. And a lot of drugs are made to be fat soluble to do that. That isn't the only way. There are also pores passing right through this fat membrane, and you can make maybe this is another drug, it's not felt soluble, but it can get through because it can use these pores. And these pores use up energy, which is what that's trying to tell us. So your cells are actually using energy to absorb that drug. And they do that for all kinds of different chemicals. And if you can get it to do that with your drug, then you've solved your problem of how to get your drug absorbed into the body of your patient. It can get right through, or sometimes there are more restrictive ways of doing it. This type of thing depends upon your genetics, and it's going to be variable to some extent. So this is the first of our barriers that can be variable from one patient to the next, because this structure if in one person might let things through easily, in another person they might have a mutation and it lets things in a little bit less easily, or won't like your particular drug as easily, and it just doesn't absorb as well. So that when we talk about having trouble with absorption, which is what interaction can be part of, then it could be one patient can absorb fine, the other patient can't absorb very well at all. So the patient that doesn't absorb very well, you might give a higher dose. So there's more of this around for them to get into their cells, but you're wasting a lot because a lot will stay outside. So even just absorbing the drug through your cell wall is complicated and fraught with variability and ability to do it from patient to patient. I think I went through a lot of this. So different methods of absorption. There's the oral into your gut, IV we talked about. You can have injections intramuscularly. We can inhale. You can squirt it under your tongue in a sublingual application where under your tongue is thinner membranes that we can get drugs through. You can nowadays have patches that you just stick to your arm, and these preparations can pass through those tough layers of skin and into your blood. So these are quite specialized, and they can be a really good way to get a nice steady flow of drug into your system without having to take lots of tablets. And of course, sort of related to that, we can also just have medications that you just spread on the top of your skin, so the topical applications. This type of thing, oral, is influenced by your stomach contents, as we know. Itraconazole, if you don't have enough acid in your stomach, it doesn't absorb very well. That's because it's not dissolving, and then once it's dissolved, it can only get into your bloodstream once it's dissolved properly. So there's another potential barrier, just the stomach contents and acidity. These are the problems. We much prefer to give patients oral drugs because, of course, it's much nicer to just take a tablet twice a day than it is to actually have to inject yourself. But all of these problems are in the way and have to be overcome before we can make a pill that can get that drug into your blood. This word, bioavailability, means how much can we get into, how well is the system I've just been talking about working? How much of the drug gets into your cells? This is different for different drugs, and we've just been through some reasons why it's different, and it's different for different people. Well, if you take an oral drug rather than IV, less than 100% will get into your blood. And this is a little graph which shows the difference. So this is, the red line is where you've taken the drug orally, and there's a concentration of the drug in your bloodstream. And it gradually rises as it's absorbed, and then it's used, and then as it's metabolized, it drops. On the other hand, if you inject it IV using a needle, you will get straight up to the top dose, and then it just gradually is metabolized again. People who are interested in bioavailability use these graphs to calculate how much your dose should be, because if we know that the drug should be about 100% bioavailability, should act like this, but this second line where they've taken the oral drug is way down here, then we know to give you a higher dose because it will be more effective to give you a higher dose. We don't want to go too high because it would start to be toxic, because this hangs around a lot longer than this. So there's complicated maths involved in using these two things. If you've ever done further maths, you can talk about area under the curve in both cases, and you can do your complicated maths and come up with the drug dose that most patients should be given, and that's how we arrive at why you should have 40 milligrams a day or whatever in the form of a tablet. There are also tablets that are slow release. No posaconazole is available on a gastro-resistant, and that avoids this peak so much, and it would smooth it out so it goes on for longer. So it's a much less up and down, which is much kinder to your cells. And we would have to say if this was converted to gastro-resistant, it would probably stop about halfway up. So we'd have to make the dose twice as much as the previous tablet to reach that level, but it would stay at that level for longer. So it would be better for your body. These things can be compensated for with dose, as I've just said. So this is another issue. If we can adjust the dose to overcome lack of a bioavailability. However, the lower the bioavailability and the more expensive the drug, the less likely we can do that, because we're wasting an awful lot of drug if it doesn't get taken up very easily. So swings and balances, as with all things in medicine, we're trying to minimize the cost and maximize the efficiency. Okay, we'll move on. We've done absorption and covered some of the problems there are with absorption. We can go on to distribution. Oral drugs, you won't be surprised, are mostly distributed via your blood. It's absorbed into your blood or your lymph and is distributed around your body in your blood serum. Because it's distributed everywhere, it gets into every part of your body, and it can, something that you want to do to one part of your body might not be something you want to do to another part, and it'll cause a side effect. So it's not very well targeted. So although they're easy to use, they are more prone to side effects. So that's a plus and a minus. Inhalers, on the other hand, you inhale directly into your airways. We can get to higher doses because we're applying it to a much smaller area, not the whole of your blood supply, just your lung volume. It's mostly restricted to your lungs, at least at first. So it doesn't get to places that you don't want it to get to. So there are fewer side effects. We can't say there are no side effects, but they're certainly reduced. And we can get higher doses in there. So if we can get it to higher dose, are they more effective? And if so, how effective, and so on. So distribution is important. The way we use it is important, is to side effects, and how prone something is to side effect influences the dose we can use. So if you can get something in through the absorption route, but you can only get in a certain amount into the target area, but the rest of your body has now got a high dose at the same time, then you get lots of side effects, and we won't be able to use that drug until we can work out how we can get more into the site that we needed to get it to without affecting the rest of your body. We sort of get this with antifungal drugs. We get and steroids. We can get lots in through your taking your tablets, but you all know the side effects that those drugs cause. If we can, on the other hand, we can keep you on, say, a steroid inhaler, and get you off oral steroids or reduce oral steroids, you'll get far less in the way of side effects. And you might, and for some people, you might be able to stay on that for some time and still adequately control your asthma. So these are why it's just so complicated. So distribution, right. So we've got absorbed, we've got it distributed around the body. So now we have to talk about a little bit about what happens once it's in your bloodstream. We have to take a step back and look at how our body defends itself against toxins. And by this we mean all environmental toxins around us. We are surrounded by nasty things that are trying to do us harm. Our first barrier to some chemical that we don't want inside, like if it was bleach. We don't want that in our bodies whatsoever. And if you spilled it on yourself, heaven forbid, your skin would protect you from that getting in your body. So the skin and actually the gut itself, just to a lesser extent, I guess, are the first barrier anything that tries to get in your body meets. And the skin and your skin and gut are good enough to get to resist most attempts to invade your body. And this toxin includes infection as well. So we know we should be all familiar about that. If it does get past our skin and gut, whatever it is, and remember that every medication we take is a chemical that our bodies don't know it's trying to do as good. They will just see it as a foreign chemical and they will treat it as a toxin. The primary function, the primary way these toxins are removed from our body is through our liver and our kidneys. I'll talk about that in a bit more detail, but those are the two organs that will recognize these toxins, tag them, break them down, and make them available to the kidneys to filter out of our bodies. And I'll go into details about how it does that in a minute. And of course, like I say, abnormal cells, infections, and so on are dealt with by our immune system. We're now going to talk about how our liver and kidneys protects us from everything. The liver is effectively our filter. Anything that shouldn't be in your body, it can recognize, and it can initiate a process that cleans it out of our blood. And that includes any drugs that we take, because they will be recognized as foreign, and our liver will say, that shouldn't be in here, let's filter it out. And a huge amount of blood passes through our livers every minute or two. So our liver is exposed to most of our blood every day, or all of our blood every day, probably several times an hour. So as it filters out, it's very efficient at filtering, because it can get at all of our blood all of the time. And if anything's in there that shouldn't be in there, it can filter it out. It is actually also, it's not all about filtration. Our livers are also very good at making nutrients that we need, using other parts of the function of the liver. I just thought I would mention, clarify, adverts that I regularly see which are commercial adverts, which claim to detoxify your liver or detoxify your body, cleanse your liver, and so on, which you'll see all over the place on the internet. Your liver is the detoxifier. Your liver is incredibly good at doing what it does. Adding in commercial, would you call them chemicals, herbs, whatever they are, doesn't really make much difference. There are a couple of instances where something you take can influence inflammation of your liver, but in talking about detox or liver cleansing is mostly an unproven thing, and you should mostly ignore it. Instead, what you should do is care for your liver. I'm speaking to all students here. Minimize alcohol intake, or when we say minimize, I suppose you would say moderate alcohol intake. Alcohol is a toxin. If you take it in, produced by a fungus, if you take it in too much every day, your liver can cope up to a point, but will start to be damaged by that. So moderate alcohol intake. I think the guidelines for the NHS were 2 drinks, which I assume means 2 units a day for women and 3 for men. So why men get to have more fun is beyond me, but maybe we need it to compensate for a stressful life. Too much weight is a problem for your liver, so try to avoid getting too heavy and avoid risky behavior, by which we mean stuff that most people wouldn't do, which would be intravenous drug taking, for example, can infect you with hepatitis C. So those are the main ways you keep your liver healthy. There's no need, as far as we can detect at the moment, for most of this stuff is just marketing. Here are all the things your liver can do. What they split it up into two phases. Now, remember I talked about fat soluble chemicals, and for toxins read chemicals. Your liver takes up fat soluble chemicals. These are things we can't get out because they can't. They're not very good at crossing the cell membrane barrier, so we have to do something with them to get rid of them. And with that is done by it goes into the liver and starts with something they call phase 1. And phase 1 can be any one of these processes. These processes are caused by enzymes. I think I've got another slide which tells you a little bit about enzymes, but in your liver is rich with something called cytochrome P450 enzymes. And those things can cause, let's to keep it simple, oxidation. And all that oxidation means is you're adding oxygen molecules onto the toxin, the chemical. In our case, it would be the chemical we call itraconazole, for example. It oxidizes it. Once it's oxidized, and to function this way, it needs these vitamins and minerals. So this is why you're taking these vitamins, B2, B3, B6, B12. One of their functions is to keep your liver doing this. So this is why these are important. Once we've got through phase 1, it passes to phase 2. Phase 2 carries out another bunch of enzymes that carry out all these different chemical processes. And what the effect of all that is, and to do all that, we need all these nutrients. So you can see there's a lot of stuff needed to keep your liver healthy. Once all this has happened, what you've converted these fat-soluble toxins into is water-soluble waste. And once it's soluble in water, it can be passed down to your kidneys and eliminated via bile. Some of it is eliminated in urine, some of it as bile and stool, depending on stuff I don't know about. I am thinking that most of the water-soluble stuff will go out in your urine, and if it's not quite as water-soluble, it might end up in bile or stool. But that's the general principle. That's what your liver's doing for you every day. Just a little short thing. What is an enzyme? Okay, an enzyme is just something that makes chemical reactions work faster and with less energy. Our bodies are full of millions of chemical reactions, and if we weren't also full of enzymes, which are just proteins, this is why you eat protein for dinner, because you need to make millions of enzymes every day. If these didn't work, then our bodies wouldn't work, because it would these chemical reactions that we need to live wouldn't go fast enough and would use far too much energy, and life wouldn't be possible. Enzymes literally make life possible, life process possible. And all they do is very simple. You've got two chemicals here, and in the yellow, we've got an enzyme, and the enzyme has two pockets, one for each substance or substrate, as they're calling it here. It fixes the two substrates together in a particular way, and then it joins them. That's all it does, and then it releases them, and now you've got a new joined up compound. So in our case, one of these would be our itraconazole, and all the cytochrome does is add oxygen onto your itraconazole molecule, and once it's oxidized, it releases it. So now what was oxygen and itraconazole is now oxidized into itraconazole. That won't dissolve in water. That will dissolve in water, and that's the important detoxification process that happens in your liver. Okay, like I've just said, pretty much what I've just said. Phase 1, cytochrome P450 enzyme, and we've talked about that a few times. I think Lauren's talked about this in the past. This essentially just converts the drug to make it more water soluble. Once it's water soluble, it can pass to phase 2, and it can be processed again and we pass it out through our kidneys, mostly through our kidneys and urine. And that happens for all azoles. It's not as easy as that. These CYP450 genes, there's at least 30 of them, 30 different genes. Each of us have a lot of variability. So you'll have some people will have more of some of these genes, some people have less of some of these genes. That's the principle of genetics. So some people will be able to metabolize faster, some people will be able to metabolize slower, some people will struggle to metabolize some things because each of these genes will metabolize a different substance, a different set of substances, and they'll be quite poor at doing that. Also, the amount of either of these genes, or the expression of these 30 genes, can vary a lot, depending on all sorts of factors, and the ones I just picked out of the large lists that I was looking at this morning. You can effectively increase the amount of these enzymes in your liver or decrease, but when you're taking a different toxin or a different drug. So now we've got another drug that we might be taking can affect the oxidation of itraconazole, which will affect the amount that we can get rid of. So if that interferes with itraconazole, then it's slowing down the amount we can get rid of. And if that happens, then the amount that stays in our blood is increased because we're not getting rid of it fast enough. And it can also happen the other way around. It can be decreased. So there can be, so you can have too much itraconazole, too little itraconazole, all due to the influence of another drug, another toxin that you might be coming into contact with. Loads of variability, and this is one of the main mechanisms by which we have to be really careful about how much of each antifungal is in your drug for you. This is personal to you. It's different for every patient. That's what makes it so complex. Phase 2 is also caused by enzymes, and there are loads of enzymes that influence phase 2, which is where we get things into water, make them water soluble, and we get rid of them. So this part is absolutely full of, some people will be able to metabolize some chemicals fast, some slowly. For example, if you've gone out and had a heavy night in the pub on Saturday night, it could be that alcohol will interfere with the breakdown of your itraconazole, and you'll end up with more itraconazole over the weekend than you thought. That's the type of interaction we're talking about. And that can, if it's just a Saturday night drink, that's fine. But if it's a drug you're taking all week alongside your itraconazole, that's not so fine, because we will have to adjust the amount of itraconazole you're taking to compensate for this happening. And the same with phase 2. These are the reasons why it's important to know how much antifungal is in your blood and what other chemicals you may be taking. Elimination is the last phase that we've covered. Most of it is antifungals mostly end up as water-soluble chemicals, and we get rid of them through kidneys and urine. But then, of course, that's another reason why there might be problems with the amount of antifungal in your blood, because there are people whose kidneys don't work particularly well, especially as they get older. So if your kidneys are not working well, you might not, the amount you can eliminate through your urine will also drop. So your levels of itraconazole may rise. So that's one example. There are lots more. So we've got to be very mindful about what other things are wrong with you when we prescribe these drugs, and the rest to eliminate by bile and feces. So, antifungals. Azoles are particularly prone to these interactions. Why are they particularly prone? Because they can interfere, they can inhibit phase 1, or they can act as a substrate for phase 1 in your liver. So it can do either. It can also act to inhibitor phase 2. So if these are being inhibited, we know that that will could easily result in you having too much in your blood serum. If they're acting as substrates of phase 1 or phase 2, it can be too much or too little, depending on how the interference is happening. essentially, they will interfere with their own drug metabolism, and they will interfere with every other toxin, the metabolism of every other toxin that your body is coming into contact with. How much that matters can depend on a huge range. It can depend on your age, on your genetics, like we've talked about. It can depend on what other illnesses that you have, like we talked about, and it could also depend on other medications. How we ever end up with the right dose, considering all that, is pretty clever, if you ask me. I don't think there's a lot more to go. What time are we on? Okay. Oh, I'm doing all right. Is everyone still awake?
Yes.
Yes.
A few people are still awake. Good. So, if your antifungal is inhibiting, a phase 1 or phase 2 inhibitor can slow down removal of the drug. If we don't take this into account, the patient can be exposed to high blood levels. But we can manage that by giving you less drug. So that's okay. As long as we're aware that this is happening, we can compensate. Likewise, if your antifungal is one of these that acts as a substrate of the CYP450, a phase 1 or a phase 2 substrate can compete with other drugs, because the other drugs will also be substrates, and it will slow down metabolism of other drugs, increasing blood levels of them as well. So you've not only got itraconazole increasing in dose, you can have these other drugs increasing in blood levels, which is not a good thing. If we don't take this into account, the patient can be exposed to high blood levels. And again, we can manage this by lowering the dose. So it's important that we get the dose right. And anyone who attends NAC will know that we spend a lot of time in the first 3-6 months getting the dose right before we leave you to take a particular antifungal drug for a long period of time. So outcomes. To sum up, absorption can vary from patient to patient. For example, stomach acidity can mess around with how much drug ends up in your blood. Metabolism varies a lot from patient to patient, thanks to their genetics, thanks to other drugs they might be taking. Elimination can vary from patient to patient, i.e. what other things that are wrong with you. Consequently, when giving antifungal azole medication, we need to check blood levels, especially for new patients. Once we know you, normally the same thing will work for you, so we don't need to give it so much attention, but we still have to monitor it, because some of this variability can change if you change your drug, a drug you're taking, and so on. Doctors need to be aware of what other medicines are being taken. Some food and herbal substances can also play a big part. Always tell your doctor if you're taking supplements, and I'll give you some examples, I think, now. Yes, the one we know about, grapefruit juice, which we kind of became aware of about 15 years ago, that specifically inhibits one of these CYP450 enzymes. It inhibits drug metabolism and elimination, and blood levels will rise of the drugs that you're taking if you're taking grapefruit juice at the same time. Of course, if you've always taken grapefruit juice for your breakfast, and we've gone through this process, and we've realized your blood levels are high, we've dropped your dose, everything's fine. What happens when you stop taking? If you stop taking grapefruit juice, we'll have to go through the whole thing again. So under the conditions that we measured you and we optimized you while taking grapefruit juice, you should probably carry on taking it. But for most people, we would say don't take grapefruit juice. Keep things constant so that your blood levels are constant. Cigarette smoke. I don't think many people in this group are exposed to too much cigarette smoke, but if they were, this cigarette smoke induces CYP450 enzyme, CYP3A4, and 6 other members of that gene family. This increases drug metabolism and elimination. And what will happen is drug levels will, instead of rising, they will now fall because you've increased metabolism of the drug. So that's something we shouldn't really indulge in if we can avoid it, or at least keep it consistent. So once we've got things steady, then if we keep doing the same thing, we'll probably, hopefully, end up with the same blood levels consistently. St. John's wort comes up occasionally. This is a herbal medication. This one we know is another inducer of members of the CYP450 gene family. It increases drug metabolism and elimination, so blood levels will fall of some other drugs that you are taking, including antifungal drugs. So not really a supplement we would like you to take, but if you must take it, tell your doctor so we can compensate with the dose down here, and then keep doing the same thing consistently. To summarize, managing stable blood levels of azole antifungals is complicated and needs expertise. What works for one patient often does not work for another. Blood levels of other drugs you are taking can be changed when taking azoles. So it's not just about the azoles, it's about everything else too. And take care with food supplements. Tell your doctor if you're taking something that they don't know about, because it can be active in this process and mess drug levels about, not just of your antifungals either. If you're at all concerned, or maybe it's a good idea to periodically check all of your medications using this antifungalinteractions.org app. If that's too complicated for you, you can of course rely on your local pharmacist. And at NAC we have a specialist pharmacist who particularly specializes in the drugs, in antifungal drugs, because of course that's what most of our patients are on. And they can be contacted for advice if you're a NAC outpatient on the MFT website.